Today we're going to talk about neurons, which are tiny, tiny cells in your body and in your brain that communicate with one another through electrical and chemical signals and allow you to perform actions, to hear sounds, to see things, to do an array of different things. So we're going to look at the anatomy of a neuron and then go into its processes. Now, if you've ever played telephone with a friend, you'll know that you whisper something into somebody else's ear, and then they'll whisper it into the person next to them, and it'll go down the line until at the very end, the last person tries to say exactly what you said in the beginning. Now, usually, what the last person has to say isn't exactly what you said in the first place. Now, neurons work the same way, except for they're a lot better at communicating this message with one another. One neuron will receive the message, then pass it along to the next neuron, and so on. Now let's look at the anatomy of a neuron and the processes that go along with receiving and sending a chemical and electrical signal. So this is a 3D model of a neuron. Here we have the dendrites that stick out from the cell body and that receive the signal at their receptors at the very end. And then here we have receptors all along the membrane of the cell body. And coming out from the cell body, this long tube is called the axon. And here we have the axon with many sodium channels all along, and these gaps in between called the nodes of Ranvier. And these sheets that cover the axon that are called myelin sheets, and this helps the signal travel about 30 times faster than it normally would. At the end of our axon, we have the axon terminal, and this is where the signal will eventually spit out its neurotransmitters, and another neuron will receive the signal. Now, the very first step is called initiation. Now, this is when the neurotransmitter hits the receptor on the dendrite. So around here we have our potassium ions and some sodium ions just floating around the cell, and we'll talk more about that in a minute. So it starts the whole process as a tiny neurotransmitter hitting the receptor of the dendrite. Let's take a closer look at the membrane of our neuron before the neurotransmitter hits the dendrite. Now you can see here that there are more potassium ions inside the cell than there are outside the cell, and there are more sodium ions outside the cell than there are inside the cell. Now, potassium ions are actually allowed to leave during this time of resting potential, or before anything happens, before the neurotransmitter hits the dendrite. Now, these are allowed to leave because the K+, or the potassium channel, is open. And the reason they would want to leave is because there are more potassium ions inside the cell than there are outside the cell. Now, they want to go with the concentration gradient. In other words, they would like to be where there are less of them. They want to spread out evenly and disperse themselves evenly. So this is called the ionic force. Now, nonetheless, there are still more potassium ions inside the cell than there are outside the cell. The voltage, or the charge inside the cell, is negative 70 millivolts, which is significantly lower or more negative than outside the cell. So when the neurotransmitter hits the dendrite, it causes a change in the membrane and the channels that are open. So as soon as the cell reaches, instead of its resting potential of negative 70 millivolts, once it reaches negative 55 millivolts, which is its threshold potential, it causes the potassium channel to close and the voltage-gated ion channel, or sodium channel, to open. Now, this allows the sodium to flood the cell. So all these sodium ions that were hanging out outside the cell are now allowed to enter. And because sodium has a positive charge, this spikes the charge in the cell from negative 55 to positive 40 millivolts. Now, sodium enters the cell because it is attracted to the negative charge, or the previous negative charge that was inside the cell, and there are less sodium inside the cell than there are outside the cell, so it is going with its concentration gradient. Now the cell wants to re-establish its resting potential, so it wants to go back to the way it was. Because there are so many positive ions inside the cell, it needs to get the sodium ions back outside the cell. But first, the K+ channel opens. So the potassium channel opens, allowing potassium to leave the cell. Now all of our potassium ions are outside the cell and our sodium ions are inside the cell. Now potassium wanted to leave because it was attracted to the negative charge outside the cell. So it left because of the ionic force. It wanted to be outside the cell. But now we have a problem. There's more sodium ions inside the cell than outside the cell, and all our K+ are outside the cell. So we need to exchange them. We need to have more potassium in the cell and our sodium outside the cell so we can reestablish this resting potential and how it was before the neurotransmitter hit the dendrite. So we do this through this sodium-potassium pump. So in order to exchange our potassium for our sodium and get our potassium back in the cell and our sodium back outside the cell so we can return to this resting potential that we were originally at, we have to use this sodium-potassium pump. Now, the sodium-potassium pump exchanges 3 sodium for every 2 potassium. So it goes here, it turns, it puts 2 inside the cell and 3 outside the cell. So eventually all the potassium will be back where it needs to be and all the sodium will be outside the cell. So here is the axon of our neuron. So in a second we're going to look at what happens when the action potential reaches the axon and what exactly is happening in that area. So for right now, these are the membranes of the axon, and here is our myelinated sheath, and this is like an insulator which allows the signal to go faster. And so we're going to see what happens when the action potential reaches the axon. So let's focus on one tiny portion of this membrane. Now the axon's membrane is similar to the membrane in the other part of the cell that we looked at, except for it is lined with lots and lots and lots of sodium channels. So when the signal hits it, then all these sodium channels open and allow the sodium to rush into the cell. So once one of these sodium channels opens, sodium is allowed in to the cell, and this signals for the other channels to open as well. Now that all these little sodium channels are open, the sodium rushes into the cell via these channels, causing the inside to spike to 40 millivolts instead of its previous negative 70 millivolts. So here is the myelinated portion of the axon. Now this portion separates the nodes, which have all the sodium channels on them. So it looks like the signal just jumps from node to node, but what is actually happening is that the sodium ions get sucked down this myelinated portion at a rate of about 30 times faster than they go through the nodes. And this is because it acts as an insulator. So these ions are getting sucked down super fast, so it almost looks like they're jumping. So now we have arrived at the very last step in this process, and that is getting the signal from one neuron to the next neuron. So what happens is once the signal reaches the axon terminal, which is the very end of this first neuron, a channel opens, and this allows for chloride ions to enter the cell. Now once this happens, it signals for neurotransmitters in vesicles, which are like little packages, to release into the synaptic cleft. Now they actually bind to the membrane, so here this channel opens, they bind to the membrane and they release their neurotransmitters into the synaptic cleft, which is this area in between the 2 neurons. Now a few things can happen to these neurotransmitters once they've been released from their vesicles into the synaptic cleft. Now some of them will bind to the dendrites, or the receptors on the dendrites of the opposite cell. Now this causes the signal to happen in the next cell. Some of them will be gobbled up or eaten by an enzyme, and this just digests them and destroys them so that they cannot bind to the receptors on the opposite cell anymore. Now some of them will get reuptaken, so they will go through this channel back into the original cell, and this allows for them to be reused by the original cell once the next action potential happens. Now lastly, some of them will land on this autoreceptor. Now this stops the signal altogether. So once they land on the autoreceptor, the signal is stopped and the process ends. We just learned a lot about how a neuron works, how it receives a signal in the form of a neurotransmitter on the receptor of a dendrite, how it travels down the neuron, and how at the very end of the neuron it's spit out into the synaptic cleft and received by the dendrite of the next neuron. So two very important things to remember about this process are, one, it is an energy-saving process. Through the ionic force and the diffusion force, which we talked about, there is very little energy required, and this allows us to constantly be using these cells without actually having to consume energy or consume food in order to drive these processes. Another thing is that the signal turns from a chemical signal in the form of a neurotransmitter to an electrical signal in the form of an action potential, and back into a chemical signal when the vesicles spit out the neurotransmitter into the synaptic cleft. So thank you for listening. I hope you learned a little bit about neurons, neurotransmitters, and the transmission of signals in our body from one neuron to the next. Thank you.